Brilliant. Firstly, thanks to Kate and Will and Willem for the invitation to speak. So I'm going to talk around some of the work that we do in the Centre for Genomic Pathogen Surveillance. So we're based at the Big Data Institute and the Pandemic Sciences Institute in the University of Oxford. It's like a whole concatenation of organisations within one particular university. But in the centre, we really focus on sort of three major tranches of work. One is a genomic epi, so understanding the pathogen dynamics of ever increasingly large genomic data sets, both in time and space. Capacity strengthening, so we run a NIHR Global Health Research Unit on genomic surveillance of AMR, and this is really focused on what are the technical support packages that are required to embed genomics into routine AMR surveillance on a national level, and then engineering and data analytics. So we have an engineering team that really has over the years built a number of web and mobile applications focused on how do you collect, merge, interpret, analyze, and present linked genomic and epi information for decision making at really a local, national, and international level. We're also, we host the WHO Collaborating Centre on Genomic Surveillance of AMR, which enables us to move towards horizontal technical support and policy for member states to be able to adapt and address surveillance systems to embrace all of the sort of work that we're focused on collectively around the value and the use of genomics. So some fairly sort of basic, obvious slides, but why do genomic surveillance? So one key component is this allows us to, with precision, map the pathogen landscape. So to identify the subsets of populations, identify where they are and where they're moving, to identify trends and dynamic changes, and then to monitor the emergence of new things, where they're going, and use that information to inform stewardship, the identification and rollout of targeted or precision diagnostics, precision interventions, and also monitor those interventions over time as the population evolves. So back to COVID. I mean, really, sort of in COVID, genomics was used for really the first time as a frontline public health tool. This tree on the left is about 30 million SARS-CoV-2 genomes, which, you know, these trees are very pretty, but they're quite uninformative on a sort of a public health scale. So the use of nomenclatures, so the development of the pangolin system by Andy and Onyo Tool, etc., really helped us talk about units, alpha, beta, omicron, etc., that allowed us to understand where those things were and where they were spreading. Now, as these trees get bigger and bigger over time, we need different ways to represent this information. This is a screenshot from the bottom figure is from the very first report that was sent to SAGE in the UK about the first few thousand cases of SARS-CoV of COVID that we sequenced the SARS-CoV-2 as part of the COG UK consortium. But this figure shows over time the proportion of different variants, and you can see that over time, you know, beginning there was a lot of mixing, and then the pandemic started to settle down into these big waves that reproduced what we know about the pathogen population. And one of the key things in the pandemic was that actually the WHO convened a panel that defined that this was the nomenclature that would be used globally, and these nicknames, alpha, beta, omicron, were given. Variants of interest, variants of concern, what were the risk, you know, mutations in the spike protein, etc. So in this figure, you can see alpha and delta, and then this little red few bars at the right-hand side were the first cases of omicron in the UK, and you know prizes for guessing what comes next in this figure. But this sort of simplicity of the information, where we have for COVID, the organism is a virus, the variant or the clonal definition is the pangolin lineages, B117, and then those nicknames. And then our risk markers, the mutations of note. This is a very transferable paradigm to bacterial resistance. So bacteria, typing schemes that are specific for each different species, you know, that takes into consideration the combination mutation, etc. MLST, there's new nomenclatures coming along that take into consideration the additional information we get from genomics, like genotyphy or strep pneumo, the GPSC cluster definitions. And then our risk markers for AMR are the determinants, the mechanisms of resistance. And of course, this is appropriate for antifungal resistance, similar thing, fungi, methods of defining the subsets of the population, and antifungal resistance determinants. So if we combine these simple bits of information with geography and time and place, it really gives us a very simple but powerful framework for understanding where variants, clones are emerging, their risk, and where they're moving, from the location within a healthcare institute, farm, the environment, up to national level, international, and ultimately global. So back to our major focus around antimicrobial resistance, and in this case, bacterial resistance. We all know these pieces of information, right? The WHO has pulled together a very nice paradigm to really sort of rein us in as to our approaches, where the bug-drug combinations are put into these three major groups. So where are we sort of currently with our global genomic data? So in the pandemic, we sequenced over 30 million SARS-CoV-2 genomes in about 3 years, an unprecedented scale and rollout of genomic surveillance, both capacity, and the capacity was embedding platforms in various different countries. But for bacteria, what we wanted to do was to have a way of indicating sort of live what the current status of our understanding of these critical high and medium groups. So as we know, the INSTC, the International Nucleotide Sequence Database Consortium, that has 3 major nodes, the ENA in Europe, DDBJ in Japan, and NCBI in the US, which mirror open genomic data. So we wanted to plug in something that would pull this information out, use QC parameters that were species specific, run the right analytics to define the risk markers, AMR mechanisms, etc., and the typing schemes, and deliver this as live situational awareness so that people can contextualize their own genomes, but also then see trends over time. And the 2 data systems that we have developed over the years, Pathogen Watch and AMR Watch. So Pathogen Watch is really focused on being able to address and use the right analytics for the right species. So it's a community-based approach. So the analytics are developed across the bioinformatics community, and they're made easy to run by throwing a genome into a web browser, having the QC indicate the quality of the genomic data, and then do the typing, do the AMR resistance determination, etc., the molecular mechanisms, and then cluster those genomes so that somebody local can put their genomes into the context of the global data set. And as we have this live and running, we can also then summarize the information on a national and international level. So AMR Watch is a platform that delivers the priority pathogens, so we can choose a particular pathogen or to look at multiple groups and then view the distribution. So this is just a snapshot of Klebsiella pneumoniae. You can see the variant distribution, and this is the AMR mechanism distribution within those variants across Europe. And this is fully interactive, so one can delve down into multiple parameters that enable us to get on top of the data that's there at the moment. Now, globally, I think we've all been very aware for a long time that we don't feel that this is, the INSTC databases are not surveillance, they're not public health focused. It's a hodgepodge of all sorts of data and metadata. But just to give a summary of where we are, this is the distribution of those priority pathogens. And you can see there's vast differences across species, but also the collection year. This is going back for the last since 2010, and you can see big spikes and then drop-offs in the time to deposition in these open data. But clearly, much of the data comes from the high-income settings. It's about 95% of the data currently that's sat in these public archives is from high-income settings. Now let's delve down and look a little bit. This is just the priority pathogens, critical group, the Enterobacteriaceae, etc. And as of today, if you inflict the species-specific QC parameters, there are just over 300,000 high-quality genomes with time and location that have been deposited in those open archives over the last 20 years. It's not fantastic, right? If we go a little bit further and look at Klebsiella pneumoniae in the African continent, as of today, we have only 2,900 high-quality genomes that have been deposited over the last 20 years. We're not doing well enough. We need to really plug in and fill in some of these gaps. And that's a huge amount of information that's required, both capacity, etc. But this is where we sort of spend our time thinking and focusing on. And of course, this is appropriate for the WHO priority fungal pathogens. This is just a readout of the numbers of genomes with complete metadata. And by complete metadata, we're only kind of currently focusing on country and date. Was that a question or a mumble?
I think it's just an unmute, David. We'll find them
and mute them. Wonderful. Thank you so much. But again, the same sort of simple parameters we're sort of applying within the fungal space. So capacity-wise, the WHO and GLASS have a really simple but quite elegant framework for describing the routes for nations to really embed surveillance around phenotypes, but also this is where we're interested in leveling genomics on top. So having a series of surveillance sites or sentinel locations, and this could be hospitals, etc., with a coordinating laboratory and a national reference laboratory that can confirm phenotypic information. And this is the framework that's used to feed information into the global antimicrobial resistance surveillance system, GLASS. So stepping back a little bit to some of the work in the previous years in Europe. So one of the things that the European Union set up many years ago was the EARSnet, European Antimicrobial Resistance Surveillance Network. Now, this was essentially built on top of this paradigm of having a national reference laboratory in sentinel hospitals to collect phenotypic information. And then these informative maps were produced on a regular basis to show the increase in resistance to bug drugs. So this is two maps, 2012, sorry, showing the proportion of invasive E. coli isolates resistant or intermediately resistant to the third generation cephalosporins. So you can see that it's a very useful way of indicating that resistance is growing. Now we need much more granularity, but this is a very was a very good framework and system that the ECDC now takes on and reports on an annual basis. So some time ago, we were involved in a project to look at can we layer genomics on top of this, the USCAPE project, which focused on carbapenem-resistant Klebsiella pneumoniae. And within the framework of European countries, each national reference laboratory identified a series of representative sentinel sites that over a 6-month period collect the first 5 resistant and first 5 susceptible isolates. And this was used to produce an indication of what are the major and minor clones that are present in Europe over that contemporary snapshot. And you can also do other things like the phenotype genotype concordance. You can look for major and minor number of SNPs between and within hospitals to indicate that Klebs is mostly spreading between hospitals in the EU. And then we did a further study to look at long-read sequencing to try and identify plasmid dynamics. And there's a couple of references there. However, one of the major things that came out of this was the leveling of capacity in countries. So actually, each nation identifying the sentinels allowed shared training on identifying carbapenem-resistant Klebsiella pneumoniae through molecular methods, and then upwards towards is there sequencing at a national or an international level. So many countries sent samples elsewhere to be sequenced. But between surveys, so there's 2 publications that are just about to come out around the 2019 structured survey, and there's a 3rd one that's just finished. So over time, this allows us to look at the ebb and flow of these major clones. So is there an increase or a decrease? Is there increased resistance or decreased resistance with precision around, for example, increased acquisition of carbapenemase genes, expansion of clones, etc. Now, the ECDC now have taken this on and are running this as a public health initiative. So this is translated from a collection of people getting together, defining the parameters into something that is now public health practice. So one of the things that came out of the first survey was that there seemed to be an increase in ST39, which triggered further investigations, triggered precision surveillance to be able to understand what the challenges were with ST39. This is an alert from ECDC around the really nasty hypervirulent ST23, which was then picked up in other countries. So PAHO, for example, in North and South America, when this alert came out, tried to identify rapidly a diagnostic that could be rolled out across the region to identify, is this particular clone, this precision, is it spreading and where? So this kind of structure, this premise of being able to define the population allows us then to trigger precision diagnostics. There are a couple of companies at least that are trying to develop vaccines against this particular ST23, and mRNA vaccine is in quite strong maturity. But again, it also points towards that ability to monitor. So Europe's really sort of leading the charge on this continental level. I'll turn a little bit now to talk about some of the capacity building work. So actually, how does this technology be embraced and embedded in low and middle income countries? This is a project we've been involved in for the last 6 or 7 years with partners in Nigeria, Colombia, India, and Philippines. And it's really a deep partnership around trying to identify what are the packages that are needed to agnostically embed in very, very different locations. Nigeria has a national CDC and a national reference laboratory, but very few functional sentinel locations, etc., etc. So the technical support packages were around lab infrastructure and protocols, DNA extraction, bioinformatics, what are the standardized processes that could be run across multiple locations, data flow, how do you link the output of genomics to the epidemiological and clinical information, and another programs of work like being able to accept funds, good financial grants practice. This is a global standard that really enables sites to be able to indicate and handshake to funders that they are capable of handling funds and disbursement, etc. There's a CID special edition that goes into a detail on each one of these if interested. This is a publication from Iruka and team where they really now leapfrogged and embedded genomics into their national reference laboratory and their framework for building additional sentinel site capacity. So how do you define the packages that are important both at a very local sentinel level all the way up to the national level? Just a very quick example from the Philippines. So the Philippines is actually one of the most advanced countries with respect to a national phenotypic surveillance program. They have it's actually 35-year phenotypic surveillance, which includes these sentinel sites, again, within this model, that on an annual basis reports proportion resistance up to the national lab, and then these annual reports are produced with lines that go up or down, indicating increased or decreased resistance. So the Philippines now have embedded in their national reference laboratory sequencing, both Illumina and Nanopore, and those standard protocols that have been developed, enable them when the sentinel sites send in information about phenotypic profiles. Firstly, this can be used to deliver the annual reports and inform local, national, and international use and upwards towards GLASS. But this also then allows for clusters of what look like outbreaks, similar resistance profiles, to be understood whether it is an outbreak. Is it something that is a particular strain? What are the mechanisms of resistance, the risk, and are those risk mechanisms delivered on a chromosome or on a plasmid? So it really enables much more precision to deliver infection control. So the Philippines have translated this into a government-funded initiative called Spot and Stop. So essentially on a regular basis, and this is every week, these clusters are identified, confirmed or denied with whole genome sequencing, so ruling in and out, and then infection control measures. So over the last year, they've identified and intervened with 13 previously unrecognized outbreaks, in this case in Klebsiella pneumoniae. So it's really, that translation into a government-funded initiative that's super important. So we're sort of, really focused on how do we sort of encourage and support scaling of this. So we put a call to action out in LID just before Christmas, really sort of focusing on some key strands that actually these are transferable technical support packages, and with the WHO really focused on how we can collectively support that across different nations. We're working very closely with the Fleming Initiative and the International Vaccine Institute and Africa CDC to host a convening meeting at the beginning of March, really focused on what are the packages and the funding that's required to take this across the African continent. If anybody is interested at all in attending, please do reach out and drop us a line. So really, sort of overall, we're very interested and supportive of defining the use cases that collectively everybody's doing at the very local level, for patient level interventions, national, for outbreaks, stewardship, etc., and then global. How do we monitor? How do we define? Turn this into public health policy rather than move from academic discovery into the public health infrastructure. And a lot of this is really about, I think Kate alluded to this, it's not really sort of technical. There's a lot of operational challenges about health system strengthening, you know, and getting better data that can feed into everything that we're all interested in around novel new methodologies. But actually, if you don't have the base fundamental good data, how can we really trust our burden estimates? How can we really trust our ability to say that we do have a good understanding of the population? And this should help us lead towards better classic preparedness. Okay, I'm going to finish there. Thank you. If there's any questions.